Welcome to the lecture series on vitamins and nutrition. Today we are going to continue with the discussion on vitamin B12, and today's discussion we are going to look into the structure. What we have already started with, we are going to describe it little more in detail, and we are also going to go into the absorption aspects of vitamin B12. Then we are going to look at the metabolic role of vitamin B12, followed by the deficiency conditions associated with vitamin B12. So, today we are going to look into the structure. As you know, in the previous sessions as we have been doing, I have been telling you that it is one of the most complex structures, and you have basically vitamins with different kinds of properties. I would say the property in terms of the substitution to the cobalt ring, this being a kind of a ligand attachment, and the cobalt link, the cobalt atom is kept in position by the pyrrole rings which surrounds, and this entire thing, what we call it like the Corin ring, is the one which is the key feature which would be there even as you could look at heme in a hemoglobin. So, heme in the hemoglobin is basically the key feature. Where you look at this particular vitamin, it is also a very interesting structure, and as you look further, you find lot of features to look at in terms of the complex formation. It is basically a coordinate complex. So, if you look at coordinate complex, we do have plenty of coordinate complexes in nature. Say for example, we have the chlorophyll, which is a coordinate compound, hemoglobin, as I have told just now. In all these coordinate complexes, you have a central atom surrounded by the ligand donors, and the central atom is stabilized, and it is one of the interesting features about coordinate complex is that there would be sharing from the ligand, and there would be like covalent bond sharing mutually, but there would be contribution of electron pair by one of the partners. So, that is the characteristic feature in any coordinate compound, and it is actually the third category. So, we are not going to deal in detail about how coordinate complexes are formed, which is beyond the scope of this lecture, but we are going to look at vitamin B12 being a coordinate vitamin, coordinate complex. As you see in the vitamin, the structure is quite interesting. So, you look over here, different forms of vitamin B12, and each form is given a name, and you have varieties of variations to the natural pattern that you see in the vitamin B12. So, you see there in nature the vitamin is mainly 2-deoxy adenosyl form, what we call it like the ADO form, what you would see normally in the mitochondria. The other natural form being the methylcobalamin, okay, which is there in the plasma. So, these two forms are quite common in the nature. There is also one more form, what we call it like the hydroxycobalamin form, okay. So, which can actually result when methyl and adenosyl cobalamin, when you expose them to sunlight or when you expose them to light, they get converted to hydroxycobalamin. In fact, hydroxycobalamin is supposed to be very active, but you find these two compounds are more predominant, the ADO form, 2-deoxy adenosyl form, and the methylcobalamin form to be very common in nature. Now, if you look at the structure, as you would see, you find the R group. It could be cyano, it could be hydroxy, it could be methyl, or it could be adenosyl cobalamin. So, this is how you call or you look at how this particular compound, especially this vitamin, can exist and perform its metabolic role. Now, if you look at the important features of the structure, you would notice that you have basically three components involved in the structure: the Corin nucleus, the nucleotide, and the second I would say covalent or the cobalt bound group, okay. So, you find over here the second group, what we call it like the cobalt bound group, where the ring tries to stabilize the porphyrin ring, which contains the cobalt, which is basically in the center, okay, and the Corin nucleus is made up of 4 reduced pyrrole nuclei, okay, and the pyrrole nuclei is linked by methylene bridges, okay, so that you can have a direct bond, and you find the cobalt being ionized and appears to be with 3 valency, okay, which is very essential for biological activity. In fact, in most cases the central metal atom in a coordinate complex, what you find in nature, whether it be magnesium or whether it be iron, so their state of oxidation is very essential to perform the biological activity. Okay. So, in this case this will also tell you how many coordinate bonds that it can form. So, in this case for cobalt you could expect 6 coordinate bonds, okay, and they are tightly bound to the 4 pyrrole nitrogen atoms. So, can you see that 1, 2, 3 and 4, the 4 pyrrole rings basically coordinate or stabilize the cobalt atom in the middle. Now, the central cobalt atom also has a provision being 6, I would say, the coordinate bonds that it is capable of forming. 4 are being formed by the pyrrole ring. So, other 2, one above and one below, you could expect there. So, that is basically in the alpha position and in the beta position. So, you find in the alpha position you can have the R group attached, and the R can be from any of this, and in the beta position you find the nucleotide. Okay. So, this is how the arrangement of structure vitamin B12 actually exists. Now, so this is one picture of how a cyanocobalamin would look like. So, you see there the cyano group on the top and the nucleotide at the bottom, and now you have the methylcobalamin with the methyl group on the top and the nucleotide on the bottom. So, can you see there in all cases the pyrrole ring is being like directly connected, and there is this bridge, methyl bridge over there linking the compounds. So, here you find a methyl bridge, here you have a methyl bridge, whereas in this range, like if you have to name the ring as A, B, C and D, A and D are directly linked. So, you do not have methyl bridge over here, whereas it is like directly linked, whereas you find a methyl bridge in all other junction like A and B, B and C and C and D, whereas A and D you find it is like directly connected. So, now we are going to look at absorption of vitamin B12. So, this vitamin B12 is also called the extrinsic factor. Okay. So, you have basically 2 ways by which the compound can be absorbed, what you call as the passive absorption, which is concentration dependent, and active absorption involves a mechanism of carrier protein. So, you see that in the passive absorption you could expect passive absorption across the buccal, duodenal and the ileal mucosa. So, it is very rapid, it means based on concentration gradient the absorption can happen, but quite not efficient. Okay. So, you see less than 1% of the oral dose is actually taken up by this route, passive absorption. So, you mean to say it is very rapid, so but there is kind of a barrier. So, we have to like explore why this particular feature happens over there. The reason is there are a lot of barrier, when being a coordinate complex, this in itself would pose a challenge for the compound to go across the barrier. Now, the normal physiologic mechanism which you could attribute for the function of vitamin B12, it is basically the absorption is through active process. So, you see that in the ileum, the small intestine, okay, you find the vitamin B12 getting absorbed. So, it starts for like few microgram, the absorption is there, although it is very small, but it is relatively very efficient. Okay. So, we have one more compound, what we call it like the intrinsic factor. So, this compound being the extrinsic factor, and you have the other compound in the gastric tract which is the intrinsic factor. So, interaction between the extrinsic factor and the intrinsic factor is basically the key that decides the absorption of vitamin B12. Okay. So, you could take this way, vitamin B12 is released from the protein complexes, okay, by enzymes in the stomach. So, proteases and other enzymes in the stomach and in the duodenum and jejunum, the small intestines, they act upon in releasing this particular compound. So, it combines quite rapidly with a protein which is a salivary glycoprotein, and you have the protein what we call as, which belongs to the family cobalamin binding proteins. You have a name for it, it is called haptocorins, because corin refers to the main ring that keeps the cobalt atom intact in the middle of the complex. So, this particular compound which interacts, which helps in the absorption of vitamin B12, it is called haptocorins. So, in the intestines, the haptocorins are digested by pancreatic trypsin, which is a protease, and the cobalamin transferred to intrinsic factor. So, you could say before the intrinsic factor could attach with the extrinsic factor, you expect this proteolytic change to happen. So, you see there the food whatever we consume, okay, so goes over here and it is broken down, digested, and in the gastrointestinal tract you have the pepsin and the acidic environment. This promotes the formation of haptocorin. The optocorin whatever you get as a result of combination, okay, goes further down and it is acted upon by trypsin. Now, in the trypsin, okay, so once it has been acted upon by trypsin, it is again trimmed further, so that it can now bind with intrinsic factor. So, the aptocorin is now degraded and vitamin B12 could find its way down, lodge in the intrinsic factor. Now, intrinsic factor bound extrinsic factor, the vitamin B12 could go across the cells. Okay, so, you have mechanism by which the liver could produce vitamin B12 and that binds to trans cobalamin, okay. So, the small intestine, and you find how this particular compound basically enters various functional compartments of the cell. So, the intrinsic factor which is produced by the gastric parietal cells, okay. So, the secretion of gastric parietal cells is accompanied by the acidic environment. So, you have the intrinsic factor cobalamin complex, in combination it passes through the ileum, and there is a specific receptor what you call it as tubulin. So, it binds to that particular receptor, and you have the brush border microvilli membrane of the enterocytes. Okay. So, here it would offer the compound to be absorbed across. So, tubulin, I would say, with its ligand IF cobalamin, intrinsic factor cobalamin complex, is endocytosed. Okay, it's mean like taken a pin side. So, the cobalamin and intrinsic factor complex enters the ileal cell where intrinsic factor is destroyed. So, intrinsic factor deficiency. So, what is intrinsic factor deficiency and how this can have an impact? Intrinsic factor is very essential for transportation of vitamin B12. So, in the absence of intrinsic factor, what happens? If there is inadequate amounts of cobalamin are absorbed, the dietary requirement is approximately 200 nanogram per day. Okay. So, what will happen? This actually results in disturbing the absorption. So, if you carefully look at it, what exactly is happening here is something like a kind of a rate limiting step. So, the amount of intrinsic factor would decide the amount of vitamin B12 that can go across after absorption. So, if you have a seal limit of 200 nanogram per day, okay, so you have the absorbed requirement should match with the body's requirement. So, if there is a mismatch, okay, what your body needs and your body is not able to get it, that would result eventually in anemia, and this category of anemia with the significant morphology is what you call it as megaloblastic anemia. So, when the root cause of the resultant megaloblastic anemia is the absence of or inadequate amounts of intrinsic factor. So, either you do not have intrinsic factor or you have intrinsic factor which is not sufficient enough to meet the needs of vitamin B12 absorption. So, this condition is what you call as pernicious anemia. So, what is pernicious anemia? Pernicious anemia is vitamin B12 deficiency, and why there is deficiency in spite of taking a diet rich in vitamin B12. If we do not have intrinsic factor in sufficient quantity, then there is a problem. The intrinsic factor will decide the amount of vitamin B12 that could go inside, and eventually even if you have more amount of vitamin B12, there is a rate determining step that prevents the amount of vitamin B12 to go across, and this leads to cumulative deficiency over a period of time, and this kind of anemia is what you call it like pernicious anemia. Okay, so now we are going to look at the transportation. So, the 3 plasma transport proteins which have been reported, one is transcobalamin 1 and 3. Okay. So, they differ only in the carbohydrate structure and are secreted by white blood cells, I mean the leukocytes. So, you have 90% of plasma vitamin B12 circulates bound to these proteins. So, only the transcobalamin 2 is capable of transporting vitamin B12 into the cells. So, here you expect how the processing could have a direct impact on vitamin B12 absorption. So, it should be circulated and then it should go across the cell. Only when it enters the cellular compartments, it would start doing its function as a coenzyme. So, you find here how or what kind of mechanism the cyanocobalamin could perform, and before that if you could look into the note of how much cobalamin is under storage. So, liver is one of the primary organs to store vitamin B12, and you could expect 2000 to 5000 micrograms are stored in liver. So, even if you lose let us say 1 to 3 microgram, the body usually has sufficient stores of vitamin B12. So, vitamin B12 deficiency develops more than 3 years after vitamin B12 absorption ceases. As long as you could have even a very minimal mechanism which keeps on topping up the storage reserves of vitamin B12. So, if you keep or retrieve vitamin B12 regularly, this could have an impact, and if the intake does not match the storage over a period, it will strike a balance and then it starts showing symptoms of anemia. In fact, the amount of vitamin B12 absorption is directly determined and by all these factors, and these factors would really give an opportunity for vitamin B12 to be steadily available in circulation, so that our body can use vitamin B12 for its essential metabolic role. Now, you look at this particular reaction. So, what is the kind of reaction vitamin B12 would help as part of the enzymes? So, it acts like a kind of a cofactor for adenosylcobalamin is the coenzyme of methyl malonyl coenzyme mutase. So, this is an enzyme that actually, if you look closely into the structure, so you see over there you have a methyl group in the middle, then you have a thiol group. So, you find here the methyl group and you find the additional methyl group. Okay. So, in this case you have L methyl malonyl succinic, here you have a succinyl CoA. See, here you have only a 3 carbon atom, but here you find 4 carbon atom. So, now you get the clue, a 3 carbon compound has become a 4 carbon compound, and here is an enzyme what you name it as methyl malonyl CoA mutase. That is quite interesting. So, a 3 carbon compound has now become a 4 carbon compound. A malonyl coenzyme ester CoA has now become a succinyl CoA in the process. Now, how do you account for? So, there should be some third party who would be providing the extra methyl group available for the reaction, and that is what exactly this particular vitamin would do in the presence of this particular enzyme. So, that way this is a very key step, because succinyl CoA, if you look at it, it is one of the key intermediate in the citric acid cycle. So, propionyl CoA is carboxylated to dimethyl malonyl CoA, so which is isomerized to L-methyl malonyl CoA, and rearrangement to yield succinyl CoA happens via the vitamin B12 dependent enzymes. So, that way a 3 carbon compound, a propionyl derivative, has become a butyl derivative. So, you get the point. This is how this extra methyl group, as you notice here, this extra methyl group source would be from vitamin B12. So, that is why vitamin B12 is considered as a methyl donor in the reaction. So, methyl malonyl CoA is formed. Now, you should ask a question, how methyl malonyl CoA is formed, and that is formed from propyl CoA, okay. And propyl CoA carboxylase is helped by biotin, vitamin B7. Now, this compound, you have the methyl malonyl CoA is formed from propyl CoA. So, propyl has only a 3 carbon atom, whereas in a methyl malonyl CoA you have 4 carbon atom, but the way the 4 carbon atoms are attached are not in a linear fashion. Okay. So, there is a pattern over here. So, you say a methyl substitution of the malonyl CoA basically results from I would say the interconversion reaction actually happens in such a way the succinyl CoA, which is a key intermediate in the citric acid cycle, would really be synthesized only in the presence of vitamin B12. So, you see there vitamin B12 is the key component that is extremely essential in preparing the succinyl CoA. So, this is how the reaction goes. So, propionyl CoA gets converted to methyl malonyl CoA, which in turn gets converted to succinyl CoA, and the succinyl CoA enters, or it is the intermediate in the citric acid cycle. So, now you know citric acid cycle is such an essential reaction, and without citric acid cycle, I mean succinyl CoA production in the cycle, you will find it very difficult for the system to maintain energy. Now, I have just picked up this citric acid cycle for your understanding. So, you see there the citric acid cycle basically starts with oxaloacetate, okay, as you see the structure here. So, oxaloacetate gets converted to citrate, okay. So, you have the acyl CoA which enters the pathway, actually adds upon the oxaloacetate to forming the citric acid. Then you have cis aconitate, then you have D-isocitrate. So, this is the transition that happens. Then you have alpha ketoglutarate, okay. So, you have a NAD, NADH H plus, okay, which acts like a cofactor, and now when you have the alpha ketoglutarate which gets converted to succinyl CoA. So, you get the point. So, any of these compounds that could be formed in outside this cycle can easily be pumped as part of the cycle to keep the cycle going. So, that way you find succinyl CoA being formed from alpha ketoglutarate. Okay. So, alpha ketoglutarate undergoes decarboxylation to give you succinyl CoA, whereas in our case if you look at how this is possible. So, this is the key. So, you have a propionyl CoA getting converted to methional CoA, which in turn converts to succinyl CoA. Now, just now we saw how succinyl CoA is one of the key ingredient in the citric acid cycle, and starting from succinyl CoA you have the reaction going further to become succinic acid, then you get the fumaric acid, then the malic acid, and again oxaloacetic acid. So, this is how the cycle continues. So, it is such a very excellent cycle for producing energy, and the arrangement of cycle is quite interesting because the succinyl-CoA can enter the cycle from various points. Now, the one which we saw, so propionic acid derivatives could be actually a wonderful source to enter the citric acid cycle in the form of what do you say, succinyl-CoA, so that it can proceed further to complete the reaction. Now, metabolic role, just now I said, so cobalamin plays a vital role in the catabolism of odd chain fatty acids. So, now this role, what we consider as a very important role, breaking down of odd chain fatty acids. Okay. Then you have threonine, methionine. So, threonine and methionine are amino acids, and branch chain amino acids leucine, isoleucine and valine. So, you see there cobalamin playing a very vital role, the vital role of catabolism of odd chain fatty acids, threonine, methionine and the branched amino acids. Okay. So, degradation of all these compounds produces the same metabolite. So, that is the key. So, you get it. So, propionyl-CoA. So, propionyl-CoA would be the starting point. So, you could consider this way, suppose if odd chain fatty acids would be part of your diet, or you could take it up as a supplement, whatever the case. So, any protein rich diet would give rise to these amino acids in abundance. Okay. So, now what you have to consider, you have to degrade these compounds to liberate energy. So, we know the mechanism of citric acid cycle and all the enzymes revolving around it, the glycolytic pathway and so on. Now, you understand how each of this amino acid can be utilized by our body. See, our body metabolism is so designed in such a way, it is prone or responds only through carbohydrate metabolism. Although we have protein and fat metabolism, you do have options for our body, and it is the most straightforward way for a body to generate energy. Okay. So, the alternate means is to have diet of only this peculiar what do you say vital substance, which actually is being broken down from the odd chain fatty acids. So, there is scope how propionyl-CoA could be formed from these degradation particles. So, you would say there is the source of protein and there is the source of fatty acid, and there is this direct source of carbohydrate acyl-CoA being formed from other sources as well. So, now we are going to quickly summarize. We are going to look at what we have done today. We have looked into the structure of cobalamin, we explained the structure of cobalamin, we looked at the way in which cobalamin could get absorbed and could reach systemic circulation. We also studied about the metabolic role of cobalamin as to how cobalamin basically acts like a methyl donor, and we also saw the role of cobalamin in the normal process of metabolism of methyl transfer reactions. So, you can take down this question. So, what is the structure? Describe the structure of cobalamin, describe the role of Corin ring and how Corin ring is stabilized by ligands. So, that is your first question. Second question, what is the metabolic role of cobalamin? What are the different types of cobalamin available in nature? And the last one being, describe the metabolic role of cobalamin in methyl transfer reactions and its role in citric acid cycle, how vitamin B12 plays a very key role in generating variety of methyl transfer reactions. Hope you enjoyed the session. We are going to continue further into different aspects of cobalamin function and its deficiency conditions in the coming session. Thank you.